wanted to give you a perspective from a user's point of view about using the resources from ARC, and specifically in the scientific area of genomics. And so I'll talk a little bit about cloud computing a little bit with respect to genomics, and then give you a taste of some of the ways that my lab has used the resources to look at gene expression using RNA sequencing, annotating microRNAs, which are non-coding RNAs, also metabarcoding data analysis, and calling sequence variants. So a lot of this work has been done by graduate students in my lab, so it's not where I've done all of these things. So I definitely appreciate their help. And so with cloud computing, this is something that is certainly recognized by the National Institutes of Health, which does fund research in my lab and other labs here at the University of Maine. And a few years ago now, they did come up with a strategic plan for data science, and recognizing clearly that the cloud is the future, right? It's a way of that you can spin up different servers that really scale with the data that you have. So it's an efficient way of using high performance computing resources. And the NIH has many different initiatives, but one of them being the STRIDES initiative, and there are others. And in terms of other efforts in genomics, certainly the NIH has several, including ANVIL and Terra and others at different NIH institutes. But the National Science Foundation, of course, has Sciverse as well as Exceed. And some of you may have used Exceed resources in the past, but a number of ways of using different cloud-based resources. And then from the commercial side, of course, we have the major providers out there, AWS, Google, and Microsoft. But indeed, they've also launched various initiatives about genomics, and actually making those genomic tools integrated into their cloud computing resources. And so this is something where there's a lot of economy of scale, just because sometimes the data sets can get extremely large. When using a cluster, I wanted to talk a little bit about, again, from the user's perspective, how you might use a Linux cluster. And if you've not done this before, the way that a cluster is organized is that there is a series of networked computers, essentially, these compute nodes. And there can be hundreds of them or thousands of them that are all networked together in one particular cluster. Then there's something called a head node, which is actually how you interface and log into the head node of the cluster to run your jobs. And the important steps in using a Linux cluster, of course, is uploading the data to the cluster, and interacting with those data on the head node. You also need to install software, and sometimes there's software that's installed on the cluster, which is great, but sometimes you need to end up installing that software in your home directory. And there's also a technology that's called containers that I'll explain. Then you have to prepare a script to actually run the software. And so those scripts will load the software on the compute nodes and actually execute the software. And there are different parameters that one needs to set. So there's a concept of a queue, which is the list of jobs. And you can configure which queue you're using, which is a series of nodes that you, perhaps they feature a particular architecture, like having lots of RAM on a particular node, or GPUs or whatever. And then you specify the number of compute nodes you want to use and the maximum runtime. And you write these scripts using in a syntax that's compatible with the Slurm network workload managers system. And that's something that's used not only at TAC, but also OSC. Then you submit your script to this, the queue and download the results. So using a Linux cluster is, really consists of these steps. And there are some really fantastic clusters. So at TAC, been primarily using the Stampede2 cluster, which is a very, very large cluster and certainly amazing statistics on its performance. And then there are 2 clusters at OSC, and Kevin showed you how to interface with OSC. But these are clusters that you can have access through Arc, and obviously these, some are larger than others, like the Stampede cluster, and it really depends on your needs. So once you have access to a cluster, there'll be some analysis workflow that you want to actually execute. So this is a workflow. Caleb Barton in my lab made this diagram. So for what we call reference-based RNA-seq analysis workflow, and using this, we can look at gene expression, say in the zebrafish, in response to something like influenza A infection. And then by analyzing the data, look at differentially expressed genes. And so one would want to run each step, as shown in these blue boxes. And so one needs to have the software installed on this Linux cluster. And you can look at all of the software that's installed using a command called module, and look at what's available. And some of the ones I've highlighted are ones that we commonly use. If the software is not accessible on the cluster, then you can install that software in your home directory, or load the software using containers. And wanted to mention with the Stampede2 cluster, there's a really useful user guide that's accessible online, and for an advanced user really is a fantastic resource. And if you're new to this, certainly reaching out to Kevin at ARC is a really, really helpful to have someone that can help you learn how to use these tools. So once you have the software installed, then you would write a script to actually execute the software. So this is the first step in this process, is trimming adapters and low quality reads with software called Rna-Seq in this particular workflow. And so one writes a Slurm script where you're declaring what queue you want to use, how many processors per node, what's the maximum runtime you want to use, other details about the job. And so this SBatch syntax is specific to Slurm, which is the job scheduling software that's used on the cluster. And then finally the actual command to execute the Rna-Seq script. And so one has to make these scripts in some way. These are just text files, but once you have them, then you would submit the job to the cluster using a command like sbatch, and that job will be submitted and given a job number, and you can monitor that job being run on the cluster. And yesterday, when I was working on these slides, there were over 2,400 different jobs running on the cluster Stampede2 at the same time. So there's tremendous capacity. I didn't have to wait for, there were plenty of nodes left over for me to use for this particular job. Another way to make software easy to install on a cluster is using a technology that commonly called containers. And it makes it easy to install software because the software itself may have lots of dependencies, and you can think of it like a series of dominoes. In order for one or a house of cards for that matter, everything is built one on top of another. So there are lots of dependencies, and one has to have sometimes a lot of different underlying software dependencies installed before you can successfully install a software package like Chime2 that I'll talk about later. And with containers, it makes it easy because all of the applications, including the dependencies, are essentially wrapped up together in a container. And using technology like Docker or Singularity, one can load a container, and then that software becomes something that you can execute. With Singularity, you can also include data with containers, and makes it just very easy and seamless to run. And another nice aspect of containers is that it makes your work reproducible, because you can store a Docker container or Singularity container, archive it, and whenever you need to, load that container up again, and you have that analysis environment exactly how it was before. Chime 2 is an analysis workflow that's used with metagenomic data, something we're using with the main eDNA EPSCoR track 1 project. And so this is where we're reading in sequence files and determining different amplicon sequence variants, and then annotating those sequence variants. And there's a series of steps. Each step, the way that we've implemented it currently, is a separate script. One could have all of the commands for the entire pipeline all together in one script. But what I wanted to bring your attention to is, with this line here, Singularity pull Docker, I am obtaining a copy of the Docker container for Chime 2. And then once that is pulled, then I'm running that. And using Singularity, one can run Docker containers. And this makes it very easy to have Chime working. I didn't have to ask a system administrator to install these packages. I'm doing it all just using the containers. And once you have that, there are different commands and different arguments that are used to run Chime, and that's what's being passed here. So Docker containers are really, really useful, and using them with Singularity makes it very, very easy. Other workflows are like calling sequence variants. Again, Kayla's been working on this, where we have a reference genome, and then sequence data from different individuals of this particular species, and we're trying to determine whether there's sequence variation near different loci and in the genome. So it becomes very easy to do this analysis. We have to create an index for the genome that we're searching. We do this just once, and this can be a slightly time-consuming step, 46 minutes or so. But once you do that, then you can run this workflow on one node for one sample, use a different node for a different sample. And if you add 100 samples, you could use 100 different nodes. And with these very large clusters, it makes it very easy. You're launching all of those jobs, submitting them to the queue all in parallel. So essentially the runtime that it takes to run one sample, you can run maybe 100 samples, as long as there are nodes accessible. So there's an economy of scale in that way. So what I've tried to do is to give you a sense of some of the high throughput sequence analysis workflows that we've implemented for RNA-seq, metagenomics, and calling sequence variants. And there is time that one has to invest to learn how to use a Linux cluster. But with the help from ARC, with Kevin in particular, one can figure out how to run things, especially if you don't have done this before. It's something where there is support, and that support is really, really valuable. You can't just read documentation alone and figure out how to do all of these steps. So the user documentation is certainly quite helpful, but it sometimes takes an expert's eye to understand completely the user documentation. Certainly it is easy to run different containers using Singularity, both at OSC and on Stampy2 at TAC. And certainly lots and lots of capacity in terms of having lots of nodes. So again, having the technical support through ARC is really, really important. So I very much appreciate the support. So wanted to acknowledge folks in my lab, as well as the folks at ARC and different funding sources, and especially the main EPSCoR office, who's arranged for allocations for that project at these resources. So I certainly appreciate all of that help. So I think I'll hand it back to Shane.
Thank you, Ben. So that completes the first 3 phases of the webinar today, and we have at least a little bit of time for questions. Actually, we have more than a little bit of time for questions. And I have a few myself that I'd like to ask after just seeing your presentation today and your delivery of your presentation for the first time. But maybe I'll get started here, and any audience members that would like to submit a question, please use the Q&A panel and we'll be monitoring that as well. So Ben, you're very unique in terms of the labs who we have worked with, in that you've had your students involved right from the beginning, is my memory. And I'm not sure if I'm remembering 1 or 2 students, because you've worked at both OSC and TAC.
Right.
But I'm just curious as to, graduate student participation using these supercomputer centers. Has that been a difficulty? What are the steps you're taking to make sure the student can be successful?
Yeah, they've certainly taken a lot of self-initiative to just have the confidence to dive in, and certainly I gave them some template scripts to get started with and things. One thing that is helpful when working as part of a team is sharing data. So we did learn how to set up groups so that we could share data amongst ourselves in the lab, and so that's something that's doable and is very helpful, because one can look at say the error log files that are generated when something doesn't work, and that's really important, is to have that accessible to one another.
Okay, thank you.
Yeah.
Now, you've been involved with ARC really from the beginning. I mean, we were speaking with you before we had these formal agreements set up with TAC or OSC, and you described to us features and attributes you thought were important. I remember back at the beginning, TAC offered container support and OSC didn't. At least that's how I'm recalling it today. But OSC more recently has introduced containers, and I'm curious, if you had an infinite supply of time on both of these providers, do you have a strong preference in terms of the work that you're conducting, your lab is conducting? And if so, why?
Yeah, containers are really important. Yeah, it's not that they didn't necessarily support them, but at TAC, what they had, and we had learned about actually through working with Kevin, is that they had the Singularity technology installed on Stampede 2, and that allows you to use any Docker. There are literally thousands of containers out there that one can access with different workflows. And one thing that I hope to do through a collaboration with the main INBRI program and the NIH is to make a series of containers that are specific to some of the workflows that I feature in different courses. And there's certainly others that have been developed for bioinformatics, but one thing that's challenging when working with in different courses and teaching people is that you can provide during the course really nice environments for people to do their work, but when they go back home to replicate that environment can be quite difficult, and containers really are the solution to that. So they can load a container and then have the exact same analysis environment as they do their work.
Do you find, the follow-up to that is, do you find that both of these centers right now are comparable in terms of your technical requirements, or is there still, in terms of your specific use case, are you leaning more in one direction than the other?
at this point, I view them as pretty equal. The just the size of Stampede 2 is very, very large and hard to get one's mind around. And so, but I think as long as the it does come down to queue times and that kind of thing, as well as the support. So I think yeah, they're both good options. And I want to say it's somewhat agnostic as to which I favor.
Well,
So
that's good, because I'm not sure that you started that way 6 months ago. So that's not
yeah, so yeah, and one thing to mention though is that TAC is one of the largest supercomputer centers in the world, right? So there are some compute tasks that can only be done on enormously large infrastructure. And so it's nice to have contacts at a major resource like that. So I think that it would be nice to keep that door open, yeah, in the future, because I could see some faculty may have needs for extremely large, yeah, compute needs.
It's our intent to keep the door open for sure.
Yeah.
So a third question I have, and I don't see any Q&A questions yet, so I'm going to keep talking. But you've spent more time working on both of these centers, and well, I guess there's one or two other faculty perhaps that are comparable. My memory is your team has perhaps interacted more with technical support at these centers than others that I can think of right
Mm-hmm.
now. In your presentation, you talked about support from ARC, and in very complimentary terms, and I'm happy to hear that. But the dedicated technical experts at these centers are very important also. In terms of your team's interactions with the support staff through their support vehicles, can you summarize your experiences? Have they been reasonably okay, or
Yeah, I think they've been good. Like with anything, having people that you can meet with in a non-pandemic world is helpful, right? I remember going to meetings with Kevin and things. So sometimes having that face-to-face contact is important. But with the help desk, certainly, there are things that come up, like filling your your disk allocation, other things where you might trigger things where really a system administrator needs to go back in and unlock your account or what have you. So there are always things where you need to have yeah, that support. But often, getting started, it might be intimidating, right, to reach out to a help desk to ask, well, can you help me write my first Slurm script, right? And there, it's certainly better to, yeah, meet with someone like Kevin. But so always having that kind of support is important, and yeah.
Okay, well, that exhausts the questions that came to my mind through your presentation. And I don't see any posted questions beyond what I've asked right there. Just give me one more second to check. I guess the only other question that I'm seeing here is why are there two supercomputer centers that we're working with, where there's quite a few in the United States, from East Coast to West Coast. So Kevin actually was very involved with the initial surveying we did. This goes back about a year. So I'm obviously a proponent of OSC because I have almost 20 years of history with them. TAC I'd never worked with, to be honest, but it was clear that they're one of the largest in the country, if not the largest. They're 8th in the world right now from their own data. So we kind of gravitated in that direction after engaging and speaking with TAC, but there are a few other attractive centers. So why aren't we already working with them? Well, we'll let the customer needs drive those discussions. If we work with a lab or a department on campus, and TAC and OSC are not suitable for the use cases at hand, it's a bowl of cherries, and we can go in multiple directions. Now that we have these new these umbrella agreements were new to these organizations. This UMaine's request was not a standard request in recent years, but now that we have this track record with these two centers, I'm confident that we can be successful with other centers in the country. So why don't I pause there? Any closing comments from the panelists? Kevin, Ben?
Look forward to working with everybody at, expanding HPC across, UMaine and the UMaine system.
Okay, well, I thank the panelists and I thank everyone who attended today and for allowing us to record. So we'll probably break this into smaller pieces and make them available through the ARC website. Thanks very much, everybody.